And also, I'm very grateful to the Turkish Physiological Society for the opportunity. It's my very first visit to Turkey, so it's fantastic. So I hope you'll let me come back in the future. But what I would like to do also with my other hat on as president of the Physiological Society of Public Ireland is also to say very much how I've enjoyed the science that I've seen so far here, and to congratulate you. I was at the session yesterday, and I thought the science was absolutely outstanding, and I can see that Turkish physiology. So I'm really pleased to actually see that. As you can tell from my accent, I'm not British. I was born on the other side of the world, although I did study in Oxford and also trained in the United States, and then came back to Oxford. But I've also very recently discovered that I've got some ancestors in Turkey. That when my ancestors immigrated to the Kingdom from Scotland and Wales, they came to this region. And so I'm related to you. Now, what I feel quite bad about is that I should be giving this talk in Turkish. So please forgive me, because I've only just discovered that we have some commonality genetically. So that's why I'm really delighted to be here. Okay. I'm going to start with a credit slide first. Because what I'm going to talk about is really just not one study, but builds on the work of a lot of trainees that I've had the privilege of hosting in my lab in Oxford over the last 30 years. And you can see from the slide that looking at the flags, it is very international. So it's Irish, German, American, Polish, Australian, New Zealand, Canadian, Chinese, South Korean, Singapore, Iceland, etc. So even though the United Kingdom has left the European Union, Oxford is not left the world in terms of academic work. So as scientists, we don't really do national borders because knowledge moves very freely across borders, and therefore that's very important. So if you're after the truth, then you have to lots of interconnectivity. And so this is one of the big philosophies of it's very open. Okay, this particular picture here is the context of what I'm about to talk about. Football fans in the audience, some of you might remember this. This is 10 years ago when the Bolton Wanderers midfielder collapsed in a Premiership game in England when they were playing Tottenham Hotspur. And luckily for him, there was a cardiologist in the stand who was actually a Spurs supporter. But he went down and resuscitated the man, so he survived. And this was a very unexpected event because he was an athlete, he was very fit, shock to the community. And of course, in the Euros the year, you saw the same thing with the Danish footballer who collapsed in that particular tournament. So, what I'd like to talk about, I'd just like to make 4 key points in the lecture. I'd like to just briefly give you the rationale for why we are studying what we're studying, and talk about the model systems that we use. So I only work on 2 cells, cardiomyocytes and sympathetic neurons. So I'd like to talk about the co-culture system, all the way through to our new work on human iPSCs as an analog system to study disease, human And then hopefully you will see from some of the genomics work the pathway or target discovery, and then how we can use these molecular highways for hypothesis generation to then test the utility of certain signaling pathways with the aim of therapeutic opportunity. So why should we study cardiovascular autonomic pathophysiological states? And I think primary cardiovascular diseases that you see, heart failure, post-MI, these are also diseases of the nervous system. And if you have this neural phenotype, then this is a very powerful negative prognostic indicator for arrhythmia and sudden cardiac death. So impaired cardiac vagus is very important in the etiology of developing arrhythmia. So the cardiac vagus, which is what we teach, is nature's calcium channel blocker. And when disease gets rid of the cardiac vagus, you get into the situation of sympathetic hyperactivity, and of course, clinically, the drug we'll cite is the beta receptor for using beta blockers for the last 40 to 50 years. Now, when we teach medical students, if you want to be a cardiologist, you want to study the heart. If you want to be a neurologist, you study the brain. But seldom do we bring these two areas together in the teaching of a doctor, because they go down their subspeciality. And of course, the heart is hardwired to the nervous system. So these two organ systems are not. We've known for a long time that the nervous system affects cardiac excitability. And in particular, patients or subjects that drop dead normally have, and these channelopathies are represented in mutations on a number of particular ion currents. So in the bottom, this is the cardiac action potential, which would be of the upstroke, which is sodium dependent, the repolarization, the plateau potential is where calcium enters the cell, and in so doing causes calcium release from the cytoplasmic reticulum. That calcium binds onto the contractile protein, troponin C, and then you get cross-bridge cycling, and that is the power stroke. The ventricle contracts, blood leaves the heart. And in order for the next phase of the cycle to take place, you must have relaxation, which is just. And for that to happen, you must close calcium channels. And the calcium channels close off because the membrane potential now moves to the repolarizing state as the potassium channels now open and potassium leaves the cell. And this repolarization is brought about predominantly by the delayed rectifier potassium channel IK. It's in two components, IKR, which is the fast component, and IKS, the slow component. And then finally, the last phase of repolarization is activation of the inward rectifier called IK1, responsible for setting up the resting membrane potential and the last phase of repolarization. Now, if you have a mutation in one of these ion channels called channel, and typically if you have a mutation in a potassium channel, this is called long QT syndrome. So long QT syndrome is very easy to diagnose because the QT interval on the ECG is long. And the most dominant form of the mutation is in IKS. This is long QT1, and the protein called MINK delayed rectifier rapid repolarization. So long QT1, long QT2 takes about 80% of the long QT syndromes. The other major mutation that you get for channelopathies is what's called CPVT. This is a catecholaminergic, and the mutation here predominantly resides in the ryanodine receptor on the SR, meaning that this channel stays open for a longer period of time when calcium-induced calcium release takes place. Diastolic calcium potential in the cell. And as the cell tries to maintain calcium homeostasis, what it wants to do is extrude calcium. It wants to push the calcium through the calcium ATPase out of the cell, and it also wants to drive the calcium back to where it came from, that is the cytoplasmic reticulum. And in doing so, it activates the phospholamban-driven circuit 2A. This is called CPVT1, and CPV2 is another mutation in calsequestrin. But I just want to focus with the experimental data in the second part of the talk on CPVT1. Now, what promotes the pathology for these ion channels is activation of the sympathetic nervous system, because these patients tend to have arrhythmia and drop dead during physical exertion and emotional stress. So as I mentioned, the heart is hardwired in, so the problem that the cardiologists and geneticists have picked up resides in the cardiac myocyte, but the trigger of this event is the nervous system. And over a number of years, we've built up a new paradigm, a signaling paradigm from our colleagues, where we have been able to identify some key proteins which are dysregulated, which affect exocytosis, that is neurotransmission in the system, coming down to the beta-1 receptor. And of course, this GPCR signals through classic phosphorylation of key proteins in the myocardium itself. And for us, what is very important for calcium homeostasis are these two cyclic nucleotides, cyclic GMP and cyclic AMP. And the two cyclic nucleotides, the efficacy of them is affected by a phosphodiesterone, particularly PDE2, which is involved in nucleotides. And it is the imbalance of the two cyclic nucleotides that sets up the dysregulation of calcium homeostasis in the neuron, promoting more neurotransmission. And you can target the signaling pathway a number of ways by interfering with the key proteins that get to the second messenger, soluble guanylate cyclase or particulate guanylate. Okay, so what we've done is researching more, but I've moved on that on that only human cell work. And what we have done in this work of a current graduate student, Dr. Nilai, who is and what we've done here is taken skin cells from patients that have a known genotype and a known phenotype, that is, that have got one of QT or CPVT. And what we've done is so we have made iPSCs, and we have then redifferentiated and then we can put the cells together so they form axonal driven synapses in co-culture. And we've done this to set it up as an analog of disease. But I'd like to go back a little bit now to give you again context and also to give credit where credit is due. just to make the point that if you can't regulate cyclic AMP in the cardiac cell or the neuron, this is a problem for you. And this is a nice study that was published in 1992 from Lionel Oakey's group in Cape Town in South. And they measured ventricular fibrillation threshold, tissue levels of cyclic AMP, and they perfused the rat heart with epinephrine. Is that as you increase the levels of adrenaline, the ventricular fibrillation threshold goes down. That is, the heart is more prone to arrhythmia. And correlated with this was a rise in tissue cyclic AMP. Now, when they prevented the hydrolysis with theophylline of the cyclic AMP, they found that they could shift the sensitivity curve to the left. That is, the heart became more prone to ventricular fibrillation. And again, that correlated with a big rise in tissue cyclic. Conversely, if you give a beta blocker, right, that is, you can delay the onset of ventricular fibrillation. James Black's discovery, the Nobel Prize-winning discovery of beta blockers, is still the mainstream current therapy given to patients to take down symptoms in the system, notwithstanding the fact that you get a lot of Now you'll see that the beta blocker was delaying the rise in cyclic AMP. Eventually, if you give enough catecholamine, the heart will succumb, because this is a competitive antagonist, that if you have enough adrenaline or sympathetic drive, you will drive the blocker off the receptor, off the beta-1 receptor. And this is what happens here. Eventually, the cyclic AMP goes up and the heart will go into cardiac arrest. It's very similar if I took a syringe of dalbutamol or isobutyrenol and I injected it into you, even if you were taking a beta blocker, eventually I would be able to tip your heart into a cardiac arrest. Okay? So whilst beta blockers are good, they are not 100% protective. This is why people still die. So this experiment here is just a lot. Just to make the point again. So here is an isolated ventricular site with a fret sensor for cyclic AMP. This is EPAC H1A. To the cardiac myocyte to measure cyclic AMP. And we've challenged the heart cell with the beta agonist isoprene. And in black, this is the dose response, and the cyclic AMP goes up. But the key point I want to make in red is that if we take a myocyte from a diseased animal, and this is an animal that has got a genetic predisposition to hypertension, it becomes hypertensive. It has got a neural phenotype and an adrenergic phenotype. And you can see here that the cyclic AMP response for a given level of ISO is much greater. Now, if you join the cells up now, so if you take the ventricular cell and you put the sympathetic neuron with it, so there's just two cells in co-culture, and if you put the fret sensor back in, and now if you come along and you activate the neuron with nicotine, or you electrically stimulate it, and you measure the cyclic AMP response, on the left-hand side, this is the increase in cyclic still in the dynamic operating range of the fluorophore. But on the right-hand side, if you take cells from the rat that has a disposition, response is that much bigger in the system. Excitability that is taking place. Now, Now, what Hayley Larson did, Dr. Larson, as part of her thesis with me, she did a little clever experiment, a very simple experiment, but she cross-cultured the cells. She asked the question, what happens if I put healthy neurons on top of disease cells? And vice versa, what happens if I put diseased neurons on healthy cells and look at the readout of cyclic AMP in the coming? Essentially, she wanted to ask the question, can the sympathetic neuron be a primary driver here? And basically what she found is that this is the control response here, this is the hyper response of cyclic AMP. When she put a healthy neuron on a diseased heart cell, she could bring the cyclic AMP response back to mimic what a normal cell would look like. And conversely, when she did the opposite experiment, she could partially recapitulate the disease phenotype in the healthy cell by having a diseased neuron present. So she made the argument, yes, the neuron is a very powerful driver of the second messenger, especially in the disease state. Mechanistic pathway linked to the cyclic AMP is also membrane excitability. That is, what is it that sets up the hyperresponsiveness from the sympathetic neuron? And these are fairly recent experiments published. And what we found here is that this ion channel called M current is downregulated in the disease. This current is important for restricting excessive firing of the neuron. So the simple comparison, in blue is a healthy neuron, and red, this is the diseased neuron. And you can see the excessive excitability of neurons. Now, we could mimic the disease in a healthy neuron if we inhibited the M. Conversely, we could rescue activated the M current. That is a therapeutic target, pharmacologically. Can we direct a targeted therapy to change membrane excitability in the nervous system? No one has done this before, but I would say, for proof of principle, this is a site that you could investigate. The voltage-gated calcium channel, we showed this a few years ago, but in essence, I just want to make a point that the CAV2.2, this is the N-type conductor, stays open much longer, so you're getting more calcium. And you can essentially block this response very well in the system. And as a consequence of all of this, you get bigger calcium transients compared to a healthy cell. And of course, bigger calcium transients are associated with more exocytosis of the neurotransmitter. So calcium is the key intermediary. That is the homeostatic point we're going to get to the rate for neurosecretory activity. And modulation of key proteins that target calcium are therapeutically very important. Now, I don't want to give you the impression that it's all one-way traffic here. It's not the case. There's bidirectional traffic and information that is moving from this is sort of one chapter from his doctoral work, his DPhil. Certain told, if you don't publish the data, it doesn't exist as far as the record is concerned. Okay? There's a lot of important information locked away in PhD theses that never sees the light of day. Because the only people that read this is the examiners and the candidate's mother. Right? They are the only people that read the thesis. So it's important for the students here, publish as you go along for the record. Okay? Even if it might be counterintuitive, data may not make sense. Someone else might make sense of it for you. Okay? Follow the data, publish the data, even if you don't understand it. Okay? Because you're doing a service to the community. Anyway, we rat cells that were diseased, they were hyperexcitable. And when he put healthy myocytes in the co-culture, you could see excitability right down. But what's really interesting, when he took the media where the healthy cells were bathed in, and he put the media onto the neuron, he could also excitability, suggesting that there could be some paracrine substance that is coming out of the myocytes, which is working back onto the neuron to regulate its excitability. Now, we don't know what that factor is. We need to do the mass spec on the media to see. And it's probably some neurotrophic factor that may be feeding back to regulate membrane excitability. Anyway, the key point is that it moves in both directions. Okay, so what are the key pathways that, target discovery? Don't focus on the detail of the slide, there's too much information, but I just put the slide up to make a point. So about 5 years ago, when transcriptomics became very popular, everyone wants to go out and do sequencing to find as many messenger RNAs as you possibly can, and we were in, like to use tech to try and answer the question. So when we bulk sequence our stellates, because single cell sequencing really wasn't available in scale back in 2016, but when we did bulk tissue and disease tissue, a whole lot of genes go up, a whole lot go down, and some don't change. And you get 5 kilometers worth of data that you've got to make sense of, right? And so you can spend your whole getting lost in this stuff. And you need some good computational physiologists and bioinformaticians to help you go. But in essence, what we found was that we could make gene ontologies pathways that we saw were around phosphodiesterases coupled to nucleotides, ion channels in membranes. And we built these ontologies, and but I think the most important thing we tried to do was to validate and provide contextualization of human tissue. That is, are we studying ratology, or are we studying a bigger enterprise? That is the question, are the transcripts we see in rat conserved in human? And for that, we took donor stellates from UCLA, from my collaborators, and these were patients that were organ transplant donors because they were brain dead and their organs were being transplanted, usually road traffic accident patients and people that have been shot in LA. Okay, a lot of guns in LA, people get, and if they're not dead, then often they get their organ. So interesting, we had the tissue from these people, and then we also had patients that were on purpose splenectomized for non-QT and CPBT. So now we could provide some validation of the rat data. So in essence, this is what Dr. Barsley and Dr. Davis did in my lab. These data sets are fully available. But I want to make a point that you're looking for your. But often with this, you can get surprised. Is it biological noise, or I'd just like to illustrate this particular point. So when we did the transcriptomic work, so dopamine beta hydroxylase, neuropeptide Y, tyrosine hydroxylase, monoamine oxidase, it's a synthetic neuron. There are lots of alpha 2 receptors, which expected because we know that that's important for. But what we were quite surprised about is that we were picking up this sort of continuous low-level expression of beta receptors on the postganglionic sympathetic neuron. And we thought, is that real or is that just well, B receptors are on the myocytes, they're not on the neurons. So we went back to this and, really to story short, we did single cell sequencing. And again, many of you are in this space, so you'll have seen these types of plots coming forward. And the key thing is, it's just not neurons. It's a very heterogeneous population of lots of immune cells, fibroblasts, endothelial cells, glial cells. It's very much a mixed bag. So the key thing is, do not assume cellular homogeneity. These tissues are very heterogeneous for different cell types. And the reason is home effect of cell-cell communication. That is, the connection of the immune system to the neuron, connection of endothelial cells to the neuron. This crosstalk is very poorly understood because neuron often focus. But in a nutshell, what we found is that we found lots of cells and immune cells too. But we saw a lot in these post-game. What do they do? Why are they there? And so, we deep sequenced all of this and got the same result. There's no worry about that. So again, this is a fairly simple experiment. Or cyclic AMP, or the protein kinase A, and it works for both senses. What Emma Bardsey found was that in black, when she stimulated the neurons with isoproterenol, the non-selective beta-agonist, there was no change in the signal form, which is exactly what the textbooks would say. Why should there be a change? Because there are no functional beta receptors there. That's true. However, in the disease tissue, if you give the beta-agonist, now you see the big signal come on. You see the big cyclic AMP protein kinase A signal, and with carbon fiber-based electrodes. I'm not going to show the data, it's all published, that you can get an increased transmission in the system. So what we think is happening is that in disease states, and as depicted compared to control, is that nature has increased the functional significance pathophysiologically of this control system. So you have now set up a positive feedback system. Pathology, on the whole, is positive feedback. It's not negative feedback. Negative feedback is homeostatic control. And to stop the positive feedback, you've got to interfere with the surgeon chopping something up. Okay, you must break the cycle of positivity in the system. So what we think is happening here is that there is an upregulation of this GPCR, which is causing more calcium load. It feeds back onto its own receptor, which drives even more transmission, as well as binding to the postsynaptic site. And because this neuron is highly excited, it's just not releasing norepinephrine. And interestingly, what we found was the rate-limiting enzyme PNNT, which is the so these neurons make a transition. It's not true. The neuron can also make epinephrine under disease state. And I think now you can probably see why this is the big info treatment states and in Italy, where they undertake bilateral stellectomy in patients that are at risk of sudden death with sympathetic-induced arrhythmias. And so if you cut the ganglia out, and this is human data, follow-up data, it's in the review down below, where we're looking here at ventricular fibrillation, VT, as initiated by an implantable defibrillator going off in the patient. That is, the ICD will activate if the patient is into VT and VF. naturally selectimized patient, they do much better than the patient that has just had one of the stellates taken out. Surgeon do it, because if you chop the stellate out, you've got to make sure you only get the cardiac branches of the stellate. Because if you don't, then you're at the risk of developing Horner syndrome. That is, you'll lose the innervation into the facial muscles, and cosmetically, the eyelids and facial muscles will droop. And surgery in young patients that have channelopathies, clearly they don't like that for cosmetic reasons. So the strategy that we've taken is more a gene therapeutic approach. That is, we don't want to cut the nerve, we just want to turn down the gain of excitability in the neurons. And we've done this with targeted therapy. So I'll just give you one example before I show you the most recent unpublished data. So we know that this pathway, nitric oxide activated cyclic GMP pathway, is impaired in the rat model of dysautonomia. There's expression, there's a decrease in the B1 subunit of cyclase, there's a decrease in cyclic GMP on the right-hand side. But importantly, in the cartoon here, I just want to make sure that you can still make NO, it's just decreased by the messenger itself is turned on, because the key scaffolding protein here, tetrahydrobiopterin, is still intact. If this had been pulled apart, then this would be a superoxide generating pathway. But it still makes NO cyclic GMP, but it's probably down by about 50% or more. And of course, because nitric oxide has an unpaired electron in. And of course, what free radicals want to do is make stable covalency. In biology, it makes stable covalency with heme groups, like myoglobin and hemoglobin, which is why it took hundreds and hundreds of years to discover, because it's hard. But its big biological action in cells is to activate soluble guanylate cyclase, to push up cyclic GMP levels. That's so what we did a number of years ago was so-called gene therapy work. I remember circulation research, and we had a broad spectrum adenovirus with a gene of interest. And I remember the mind back saying, well, yeah, it's all very nice, but you don't know which cells you're putting the virus into, and you could be, causing lots of abnormal paracrine. Go away and do targeted gene therapy. Of course, no one in the world could do this at this time. So I thought Dr. Markheim was being quite hard on us, actually, inserting here a run of papers in Nature at the time using the same technology. But having said that, he probably did us a favour, because with the virologists in the group, we beat it away for a couple of years, and we like this is pretty standard now, you can get all the stuff done commercially. But in 2002, 20 years ago, when we were doing this, you had to do it yourself. Okay? So you went through quite a few postdocs in virology to get this to work. But in the end, we kind of did it. So we made a specific promoter tagged with plus a report protein, either GMP or in cherry, with the empty vector. And this just wraps it up in a nutshell for you. What we found, top left panel, these are Th positive neurons. This is our gene of interest. NOS were put into only the sympathetic neuron. There's no leakage of the reporter gene out into other cell types. So that was good. We could. And then when we phenotype cells, we could take down the calcium transients. Soluble guanylate cyclase. So we could build a model whereby we overexpress this target protein to activate more bioavailability. The cyclic GMP would activate PE2, and phosphodiesterase 2 is really interesting. It's what it does here is that it actually breaks down cyclic AMP. So you get less cyclic AMP protein kinase of the N-type calcium conductors, meaning less calcium, less neurotransmission. Yeah, I'll start to speak up. So we're also very much interested in how NO works in the system. And this adaptive protein was first discovered in the brain, but it was then subsequently picked up in the heart. And this work was a really nice study undertaken at Johns Hopkins in 2008. And their motivation for doing this study is that when they were looking at the SNPs in long QT syndrome, the only QT was the mutation in CAPON, the adaptive protein. And they reasoned that if they overexpress this particular adaptive protein in myocyte, and they measured the action potential duration, they shortened it, and the answer is yes, that is, the CAPON overexpression would make more NO, and it would inhibit the calcium channel and activate the repolarization. And of course, if you get a fast repolarization, we wanted to ask the question, since it was but more interestingly for us, it is massively done. Is the increase in calcium current and calcium transient, is it somehow related to dysregulation of this adaptive protein? So we went away and we made another viral vector, this time with the CAPON, LOS1 AP, with N-Cherry to drive gene expression. And in a nutshell, what we found is that, yes, you can increase loss activity. So therapeutically, there may be two sites where you could put your So let me just finish with some unpublished data. And this is the CPBT work. And as I mentioned, CPBT is very interesting because the heart's no change in ECG. And the prevalence of this is about 1 in 5,000 to 1 in 10,000. Sadly, it's got pretty high mortality in the under 40s, so 30 to 50% die. Their first presentation is death, like Mohammed, football, right? You don't know if you've got the mutation, it's expressed, and you get a phenotypic response. So again, the main mutation is on RYR2, and CBB2 is on the calsequestrin in. So what we have done is essentially also decide in the nervous system, this one's being cardiac mutation. Because polymorphic ventricular tachycardias, which are degenerative ventricular fibrillation. The structural biology of this is really well understood now in terms of where the key sites are for the mutation. And what we have done is to look at control. And when we first looked at the myocytes, structural differences at all between the healthy cells and the so-called disease cells. When we looked at the neurons, just looking but when we phenotype cells for calcium and put it into the myocytes, I think you can see quite clearly that when we stimulate the myocyte with you can see the disease cells normal calcium regulation in those cells, which is this has been shown extensively before. This is the new result here. So you're the first one. But I think we can say with some confidence that CBBT is also a disease of the nerve. And what we're seeing here is that the blue is exposed to, they may have more firing because we have current plants. And I think the key point to make here is that they have got an abnormal calcium profile. And where we are at the moment in targeted pathways to the gene transfer sequencing of the human iPS. So, we've got miles of data here, and so it looks like there's going to be some key protein pathways to target further down the line. So I'd just like to finish now. Thank you all for your attention in this. I'd like to acknowledge the British Heart Foundation, who also Wellcome Trust, who have paid for a lot of my research over the years. This is the last time my group was together. This was the Christmas party. Famous for where William Harvey was, for those of you that are in the cardiac, 1642. So hopefully we can get back together this year, hosting my visit.
Questions for David Paterson? Ella, would you like to go first? Okay, microphone a little bit.
David, thanks a lot for the talk. So as you said, there's some immune cell involvement in this stellate ganglion, right? So what is going on with this immune cell? Or what's the role?
Yeah, that's a very good question. We actually out of Rockefeller, she's in my department. So what we just published this earlier in the we see are really elevated in these cell models. So we think this macrophage certainly affect all the calcium dynamics in the system. So I think within the ganglia itself, those and if you lose the normal regulation of them, they can be involved in early inflammation that takes place. A sensor, like a threat sensor or a calcium indicator in one cell, you've got a patch electrode down on the other cell, and you're trying to voltage time current that cell for a readout, plus look at real-time fluorescence. If you introduce a third cell, which technically could be done in a 3-cell culture model, is trying to get one variable to change whilst you're measuring another variable, because a lot of confounding information coming in. But some labs to do. If we can't do it, others will. As an early inflammatory model.
Thank you for this great talk. It's very impressive work. I have a question about this calcium, the calcium uptake alterations. I saw in your single cell sequencing there were astrocytes as well, and astrocytes are known for regulating the calcium. So, a role for astrocyte, and is that something that's...
Yeah, I think glial biology, I think the astrocyte glial interactions in the peripheral nervous system, especially in the heart, hardly anything known about that. And obviously they mop up a lot of potassium given their transport systems. But my instinct, given all of that cellular heterogeneity for one ganglia, that's there for a reason. What are those cells doing there? What is their biology? What is their physiology? So my instinct, just like the immune cell, it won't surprise me if you've got these complex parachromatic signaling systems that are involved in the overall homeostasis of the neuron itself. Cells. Again, this data. So I think these are rich for young scientists to go in, given that we've got the spatial organization of those cell types.
So, your model with the iPSCs, I think is very nice to study. I was wondering, have you think about like an organoid system that is more 3D?
Yeah.
I think people do it in the brain, I know,
Yeah.
but for the heart.
Yeah, no, the organoids haven't, the organoids done in the heart, because not many people work on the all the neural people are in the brain, the heart people are in the heart. It's that bit in the middle, which is to other people around the world. Okay. So neurocardiology is now becoming a subspecialty in science, but 25 years ago, 30 years ago, no one was cardiologists, neurologists, was not proper neuroscience. But I think now with technology, there's more scope here to open this area up. Yeah.
Like an embryonic line or a love line?
No, they're not at all black. But this is one of the big challenges in iPSC cardiac mass biology, that we can't get full maturity of cells, so they're still in their fetal state to an extent. So we keep cells to about day 20, day 25. So, and of course, we know that ventricular cells don't spontaneously make cells. If you try and mature them even further, then you lose the efficacy of the cell. The neurons are much better, actually. Now, interestingly, if you co-culture the neurons, mastocytes live longer.
I love infectious disease, so I'm quite interested in my body's like stuff. So, would you comment on like how viral infection induces us? Function and how to relate to it?
Yeah, well, my lab is really interesting in terms of inflammation. When the SARS-CoV-2, is attacking the body, at heart in the form of so you get an immune response to that, you get a dilation But if you have too much of an immune response, this increased immunity response really moves itself into sadly die of COVID, die very similarly to what you see in sepsis. That is, they die due to oxygen. So you've lost control of the blood flow, and they become hypertensive, which is why they
Thank you for your exciting speech. I was wondering that there's a remodeling. While you were answering previous question with non exercise, I understood, I think. Yeah, I was wondering the mechanism.
what we've got is something that's more phenomenological. You know, if you have high sympathetic, which is associated with exercise, then you have that's what we would argue. It's like the diagram I had of the bulb and the fuse. And what we would argue is that it's very difficult to treat the mutation of the heart. You could probably do it with CRISPR technology going forward. I think that would chop the nerves, or you could be a block. That is taking place. So for athletes, especially non-PT athletes, that you can pick this up if you have normal screening programs. That's easy. The ones I'd be more worried about are CPBTs because their hearts look normal, and unless you've got the family history footprint of those patients, they don't know they've got the mutation, but they're just carrying the mutation around with them. And I think what's really interesting is, why is it that children don't drop dead when they play? So they've had this mutation since the risk is really only seen in the late teens, early 20s, is when you get this. You don't see it in a 5-year-old or a 10-year-old, but they've got the mutation, and they're very active. So that's interesting, I think.
And it brings us to the end of this session. David, thank you very much again for coming and sharing your exciting research with us. Right after I present this society glass to David as a memory from the land of his ancestors, right after David, this is a physiological class from the Turkish Physiology Society.